Yeah, should we start?
Yes,
Yes,
yes, of
please.
course.
Oh, okay. Assalamu alaikum, everyone. Good evening. Uh, I hope you all are doing well. We are pleased to have you in this international webinar, The Future of Antiviral Medicine. This webinar focuses on the future of antiviral medicine, innovations beyond drug resistance. And thank you so much for joining. Let me introduce myself. I am Mohammad Shahzad Zafar, a doctoral research in biomedical engineering medicine at the School of Biomedical Engineering, Shenzhen University, China. My research focuses on identifying diagnostic and prognostic biomarkers in colorectal cancer and liver metastases. Beside my academic and research pursuits, I serve as the Senior Vice President of Vitanova International Alliance for Sciences, an organization empowering individuals to excel in the world of sciences by supporting progressive research, fostering collaboration, and providing specialized scholarship guidance. Vita Nova believes in the transformative power of knowledge and opportunities, aiming to create a future where passion for innovation is limitless. Let me introduce Dr. Mouna Aqerrout, the guest speaker. Dr. Mouna, she is an adjunct lecturer at the Faculty of Science and Technology of Mohammedia, affiliated to University Hassan II of Casablanca, Morocco, and a researcher in oncovirology and molecular biology with a strong focus on non-invasive biomarker discovery in bladder cancer. She has a good experience in experimental and bioinformatics-based research, working with clinical samples. Her academic background is rooted in virology, molecular biology, immunology, and biomedical research. Ma'am, come forward to deliver the speech. Thank you so much for giving your quality time to this international webinar.
Of course, thank you so much for this presentation. So, I want to thank uh, firsthand the Vitanova International Alliance for Sciences for giving me this platform to present myself as well as this wonderful subject of today's webinar, which is the future of antiviral medicine, innovation beyond drug resistance. And thank you, Mr. Medic, for presenting me and hosting this webinar. So I'm hoping everyone can hear me clear because I'm going to start right now. So can everyone hear me clear? And can
Yes.
you see the
Yes, you are clear.
presentation? Thank you. Okay. Okay, great. Good afternoon, everyone. Thank you for being here. Today, I will be presenting the subject of the future of antiviral medicine, focusing on how recent innovations are helping us move beyond one of the most persistent challenges in the field of drug resistance. So, throughout this talk, I will highlight not only the limitations of current therapies, but also the emerging strategies that are reshaping how we approach viral infections. So, before starting the presentation, I will introduce four axes that will be the main axes that are going to evolve around my presentation. So, the first axe is the challenge that we're facing um, with the antiviral drug resistance regarding different types of viruses. Second axe will be on the limitations of conventional antivirals. The third axe will be on the emerging innovations in antiviral strategies. And the final axe will be around the future, which is the predictive and the personalized medicine. Okay, so first, I'm going to introduce the subject of today's presentation, and also the problem and the challenge that we are facing nowadays. So viruses remain a major global health concern, from chronic infections such as HIV, for example, to recurrent threats such as the influenza, which is also a very, very frequent virus, and more recently, emerging viruses such as SARS-CoV-2, viral diseases continue to challenge healthcare systems worldwide, as we saw nowadays or in recent years for the epidemic of the coronavirus. So, over the past decades, antiviral drugs have significantly improved patient outcomes and survival rates. However, despite these advances, the long term effectiveness is increasingly compromised and increasingly slowing down. This brings us to the central issue of my presentation today, which is the antiviral drug resistance. So, what exactly makes resistance such a critical problem? So, drug resistance is not simply a side effect of treatment, it is a direct consequence of viral evolution. So many viruses, particularly RNA viruses, which are the most dangerous type or kind of viruses, they replicate with very low fidelity, meaning that mutations occur frequently during replication, which is the process of the viruses and how they multiply. So when antiviral drugs are introduced or induced in our bodies, they create a selective pressure. Under this pressure, viral variants that happen to carry resistance mutations gain a survival advantage. So they're ahead of us, of our biology and our genetic, and rapidly become dominant. Importantly, resistant variants of viruses often already exist within the viral population, which is against the myth or the false information of the treatment or the medication creates this resistance. It already exists. So the resistant variants, they are already existing within the viral population before the treatment even starts. So in reality, treatment does not create resistance. It has a selectivity. So it selects for the resistance. So to understand this better, we need to look at the underlying mechanisms. So viruses actually they exist. So these mechanisms of resistance, they're diverse, and viruses exist as highly dynamic populations. So we're not facing just one virus or one type of virus. We're facing multiple subtypes. For example, the HPV virus. When it enters the body. So we have the I think it's around 154 subtype of HPV that are nowadays existing. Some of them are high risk, some are low risk. But when you're infected with HPV, you don't just have just HPV1, for example. You have multiple HPV subtypes that are infecting your body. So they exist in dynamic populations, meaning that within a single infected individual, there is not just one viral genotype, but a whole spectrum of closely related variants. So this diversity, which is another mechanism, so this diversity allows the virus to adapt extremely quickly. Of course, viral replication cycles are very short. That's another addition. So allowing multiple generations and multiple viruses to replicate and generate and occurring within a short frame of time. So it's very very fast, the multiplication process and the replication of viruses, which adds to the challenge. So altogether, this creates a system where evolution is happening continuously and rapidly, as I already said, making viral infections particularly difficult to control. So this is the main challenge: is to control this infection, control this viral infection and this viral multiplication, or specifically replication. So in this sense, we are not targeting a static pathogen. As we said already, it is very, very dynamic and changing, like in a very short span of time, but rather, an evolving system. So we're targeting not just one pathogen, not just one virus, but an evolving system. This leads us to, the limitation. Now, why these drugs, why these treatments don't work, or are less effective, or are sometimes just not the right ones. Their effectiveness is faced with resistant resistance resistant force from the virus. So most conventional antiviral drugs target viral proteins. So we have these drugs that are that are made or that are produced to target viral proteins. For example, we have the polymerases or the proteases, which are known proteins that are genuinely targeted and the most frequently targeted by these antiviral drugs. So while this approach can be effective initially, so it's the first step, the to go step, generally when we get infected. So we take an antiviral. These viral components are highly prone to mutation, which facilitates resistance. And therefore, many antivirals are highly specific to a single virus. So we have also this limitation of the narrow spectrum. So we have these anti-drugs or antiviral drugs that are very specific to one single type of viruses, and they're not broad spectrum. So this is another limitation of their usefulness in the face of these emerging pathogens. Some treatments also can come with toxicity, another limitation, and can be harmless to the organism and to the body. So particularly when we use them for a long term. So taken together, these limitations, they highlight the need for new strategies. And generally, we're talking, artificial intelligence, technologies, or just new treatments adapted to a broad spectrum of viruses, which leads us to the new paradigm shift. So in response to these challenges, antiviral research is undergoing a major paradigm shift. So researchers around the world, scientists, they they rather than focusing just on the virus, they came up with new ideas, new perspectives that involves new approaches that aim to target host factors. For example, integrate advanced technologies, design therapies that are inherently more robust to resistance. So in other words, instead of constantly reacting to viral mutations, so as in the past, we were just scared and concerned of these mutations. Like, what do they bring? Like, how can they affect us? In what way? Like, what can we get from these mutations? Is this like gonna be engraved in my genetic systems, and what if, for example, for the such as COVID-19, like what if this variant is not the only one? What if it keeps mutating over time and become more robust and like, for example, like kill thousands and thousands of people and be just the most dangerous virus ever, just for example, also the HIV. Like people are scared of HIV. They're scared of this SARS-CoV-2 virus. There's the Ebola virus. There are multiple viruses that are very, very high risk. HPV as well. So we're scared of these diseases that are the outcome of these notorious viruses. So in other words, instead of constantly reacting to viral mutations, we are seeking new approaches and anticipating to outmaneuver these viruses and be ahead of them and predict them and predict their next move. So this takes us to the host target antivirals. So we're shifting from the mutations and a focus on mutations to targeting the host targeted antivirals. So one of the key strategies is targeting the host pathways. So viruses rely heavily on host cellular machinery to replicate, as we know, by disrupting these host dependent processes. So we are acting upon the inhibiting of viral replication without targeting or without harming our organism and targeting the virus itself specifically. So since the host genes are far more stable than viral genomes, this significantly reduces the likelihood of resistance. So it's actually a very good approach and not harmless to the host. We just need to be more specific with the targeting, and the host genes, knowing that they are more or far more stable than viral genomes, this reduces the resistance. So we can actually beat the virus and without harming the the um, the organism, or in our case, our body. So, however, the challenge lies in identifying targets. So there's always a problem with this strategy because it needs to be specific and sensitive at the same time. So the challenge lies in identifying targets that are essential for the virus, but not critical for the host to minimize toxicity, of course. So we said we cannot harm the host. So we have to be specific in this strategy. So we have to identify the targets, or the real targets, the most specific ones that are essential for the virus within the host. So it's still a little bit tricky, but it's a under the works, and a strategy that is very, very effective if done properly, of course. Moving on to another therapy, another approach, which is the RNA-based therapies. RNA-based therapies, they're very, very precise. We're talking about RNA. It's something that's very, very precise, very fragile. Uh, it's very, it's also very, um, very. We're talking about microscopic level of. We're talking about cRNAs, microRNAs. So these molecules are very, very small and very, very fragile. So if we can actually target these molecules in a precise kind of way, absolutely, we can specify, or we can specifically target this viral RNA and its sequences, and we can prevent detranslation, which is key point in the RNA-based therapies. So we prevent the proteins to be shaped. So we stop or we block the translation part into the proteins. So we have no viral proteins that are produced. It's one of the most major advances, and this approach is very adaptable. It's very, very highly precise. So these RNA sequences can be very rapidly designed in response to viral mutations. So it has to be very rapid to act upon this infection and to identify the viral RNA in a very short period of time, making this strategy particularly valuable and rapid. However, the challenge within that lies within this therapy, uh, efficient delivery and stability, they remain a key challenge. The delivery of the cRNAs, microRNAs, it has to be very very specific. It has to be stable, as we're going to talk later about the nanotechnology, which brings us to the notion of delivery that we're going to bring up later. Yeah, this was it for the RNA-based therapies. Moving on, we have the CRISPR gene editing. So we all know CRISPR gene editing as one of the high-end technologies nowadays. They take this concept of antiviral resistance uh, very, very seriously, and it takes it even further, like apart from the other strategies that we just saw. So instead of simply blocking the viral replication, the principle of CRISPR and the CRISPR systems uh, that they can be engineered to directly target and cleave viral genomes. So we're talking about personalization and personalized medicine and treatment. So the CRISPR does a great job into evaluating the whole viral system and organization and the population as well. So you are free to do whatever you want with CRISPR because you edit the and you engineer directly according to the target that you have picked, and you cleave the viral genome using this editing. So this is especially promising for chronic infections. So we're talking about chronic infections, chronic viruses. So these viruses, as we know, they can persist in latent forms, which is the go-to mechanism for most viruses, is to stay persistent and latent. So um, in the host cells, of course. So CRISPR could allow us to eliminate these hidden viral reservoirs. So as we know, we're all infected with viruses. Like as we speak, as I speak, and you're hearing this presentation, you're attending this presentation. Uh, I am 100% sure that every one of us right now, we are infected with viruses, different kinds of viruses. And they may not be harmless, depending, of course, on different cases, but they are hidden. They're not in action. They are latent. So CRISPR would allow us to eliminate using the CRISPR gene editing uh, to eliminate these reservoirs, these carriers, these viral pathogens that are hidden in our bodies, in our organs, in our tissues, moving from disease management towards potential cures. So it's very, very advantageous and a very strong opponent for the antiviral strategy. Although still in development, it's still in the works. Scientists and virologists around the world are still figuring this CRISPR gene editing out and using it against the different viruses. But research is still in the works and it's developing very fast. This represents, of course, one of the most transformative directions in antiviral therapy. Another strategy to fight, of course, the antiviral or virus resistance is the monoclonal antibodies. So it's it's a type of immunotherapy, of course. Monoclonal antibodies, they provide another powerful approach by directly neutralizing viral particles. So as you can see in the figure, just on the right, they bind to specific viral epitopes. As we can see, there's the host cell, there's the receptors. So they bind to specific viral epitopes, dependent on the virus, of course, preventing entry into the host cell. So they do an incredible work, these antibodies, by stopping or by blocking the first step of the viral cycle or the viral replication, which is the entry. Injury is the first step, as I said, and it's something that the virus has to do in order to have its progeneity or its the variants and to to multiply. So these monoclonal antibodies, they do an incredible job defending the host cell. Um, so it is very, very promising. It is a very promising strategy, and it's a therapy that have shown great efficacy in diseases such as COVID-19 and RSV. So yeah, we developed monoclonal antibodies based vaccines to combat the COVID-19, the SARS-CoV-2 virus. Uh, However, they're highly costly. So this is the one, I think, the only limitation that this therapy has. So they're high cost, and it is sensitive to viral mutations also, and this limits their widespread application. So yeah, although it's important and all of that, they're still not really they can't be used for, routinely therapy and routine treatment of infected patients. Moving on to the broad spectrum antivirals, another and a major goal in the field of antiviral resistance is the development of broad spectrum antivirals. As you can see in the figure, we have multiple types of antivirals, broad spectrum ones, which are, I think, they are in red. So there are multiple major. Uh, they're targeting generally. They're produced to be shared or to target conserved viral processes that are shared across multiple virus families. So they are targeting these conserved viral processes that we can find, for example, in HPV, we can find in Ebola virus, we can find in Zika virus, etc. So yeah, we find the common point between these viruses, and we can attack them at once. So this makes the virus or the viruses included very weak at a point, and make them particularly valuable in the context of emerging infections. So these antivirals generally, they're very useful, very advantageous, and rapid in terms of response, because as you use multiple broad spectrum antivirals at once, we have a better chance, a higher chance of combating those viruses than using just one narrow spectrum antiviral that can, attack or target just one virus at a time. So nanotechnology, I've brought it up earlier in, I think, as I was talking about the delivery. So nanotechnology, it enhances antiviral therapy primarily through improved drug delivery. So the delivery is the key point in nanotechnology. It's a very, very important technology that brings the notion of delivery and nanocarriers. We have, as you can see in the figure, different types of nanocarriers. We have the metal nanoparticle, as we can see that the antiviral drug is transported along this metal nanoparticle. We have the polymeric nanoparticle, nanostructured lipid carrier. Sorry, and as you can see, we have two types of lipid. We have the liquid, the solid, dependent on, of course, the antiviral drug that we are carrying. Is it going to be RNA, DNA based? Is it going to be protein based, etc.? So uh, nanocarriers can protect fragile molecules like RNA. So we already know that RNA is a very fragile molecule. It's very, very small, and yeah, it improved, of course, targeting to specific tissues, and it reduces systemic toxicity. Uh, we have to carefully pick these carriers, of course, regarding the transportation of the said antiviral drug to reduce the toxicity, of course. So this is especially important for next generation therapies that require precise delivery systems, because there are some particles that we have to really pick precisely the delivery system so that our therapy can be a success afterwards and not fail, just at the start, middle, or can even, the nanocarriers can even carry or deliver the antiviral drug correctly or successfully. So moving on to the talk of the recent research and science technologies and new approaches, which is AI, in different fields, not in just the health and scientific research field. We have AI in antiviral drug discovery. So, as we all know, AI is a revolution, and it has revolutionized healthcare in general, research also, and it's revolutionizing also how we discover and design antiviral drugs, which leads us to its application and its help and contribution into this discovery. Uh, AI, it allows us to rapidly analyze massive data sets. So this is a key point of the AI use in terms of the antiviral drug discovery. So we can do the analysis. Sorry, we can do the processing. We can do everything. We combine this data. So we're left with a lot of data. AI is going to intervene and it helps you depict this data, organize it. It helps also to rapidly analyze it, and also goes even further to prediction. Prediction throughout AI techniques, different types of uh, uh, ways to analyze, and the at a very very short period of time, and it predicts protein structures throughout different softwares and different AI based websites that are conceptualized for these tasks, and to identify potential drug candidates in a fraction of time that is required by traditional methods. So we cannot use traditional methods and cannot rely on them to make our life easier when it comes to antiviral drug discovery, especially in our time where the it is critical to elongate the research, and to, for example, to just make a lot of time and just waste a lot of time into organizing data and analyzing data that can take months, sorry, and even not even going further with the outcome of this data and what it's going to give you. Is it going to predict you the right drug? I don't think so. Is it going to help you identify a 3D molecule that is, for example, the drug target, or show you its structure, and just and helps you see the big picture and the candidate drugs that are the outcome of the first compounds. So yeah, it significantly accelerates the transition from research to clinical application, which makes it an ideal method, an ideal approach into the antiviral drug discovery. So it is a must nowadays to use AI, and yeah, to gain profit from it, and to help analyze your data and get the right and correct outcome in a short period of time. So yeah, so Moving on to the viral evolution prediction, we've talked about the AI that can, help us discover and design antiviral drugs. Now we're gonna see the AI beyond drug discovery. AI also enables us to predict viral evolution. So viral evolution is very, very, it's a complex term, but AI simplifies it. It simplifies it by analyzing mutation patterns and evolutionary dynamics, so we can easily anticipate which variants are likely to emerge. So AI lets us see through the virus and see through its evolution. This shifts the approach entirely from reactive to proactive. So we're not just reacting to mutations, we're taking actions upon these mutations as we see further, as we surpass the following of the virus. We predict um, what's it gonna do, like what is it gonna target, etc., allowing us to design therapies that remain effective even as the virus evolves. So we are going hand in hand with the virus, if not surpassing it. So yeah, it's very revolutionary. It's something that's very, it's in the works also. It's very complex, and I think this prediction of viral evolution can lead us to definitely to a better control of the viral replication of viral cycles, as in the future terms, of course. So we move to combination therapy, another type of course, antiviral resistance strategy, which is the combination of different therapies. Combination therapy remains a cornerstone in the fight against resistance, as we already know, to fight different types of, let's say, there are bacteria, there's viruses, or the outcome even fight diseases. We can include different types of therapies, different types of approaches, technologies even. So by targeting multiple pathways simultaneously, at the same time, we reduce the likelihood that the virus can adapt. So just like we combat the virus, we are just kicking the virus out of our body, in other terms, is we reduce its likelihood of adaptation or of adaptation. So this strategy has been particularly successful in HIV treatment. As we know, the HIV patients, or infected patients, they take different types of antivirals, and they undergo different types of treatment and therapy. So, yeah, it will continue to play a central role in future therapies. It is a very important type of therapy. Sometimes it's the only way the therapy can work, especially if we have a very high risk type of virus and very dangerous type of viral infection. Moving on to the immunotherapy. It is also a high end therapy, very costly, as we know. It's a therapy that focuses on enhancing the host immune response. So talking about the uh, immune system of the host and enhancing it. So approaches such as interference and immune modulators, they can help our body control these virus infections or these viral infections with a more effective way. So there are different types of immunotherapies we can see here. Using cytokines, the use of cytokines, the use of CAR T cell therapy, which is very costly. It is adapted, it is applicable in clinically, but it's very costly. Checkpoint inhibitors, very very niche subjects of research in terms of antiviral strategies. Vaccines also, they're very critical. They're very in terms of research also, but they demand a combination of different I wouldn't say sciences, but just different fields, because we're talking, for example, when we say the HPV infection, for example, and its vaccines, as we figure out over time that the monoclonal vaccine, it's not working anymore. I mean, it had never worked. As you can see, we use nowadays the poly vaccines that can target different subtypes. So vaccines is very sensitive. You have to gather the molecular biology, there's immunology, there's even there's genetic, epigenetic, etc. Different, very different aspects and fields of study. So yeah, so besides immunotherapy, or just inside or in the immunotherapy world, vaccines, they're generally the first way of prevention against viruses. So they remain one of the most powerful tools in antiviral medicine. They're the first prevention system. They can never get outdated. So the development of mRNA vaccines, it has demonstrated how quickly we can respond to emerging threats. So yeah, they are just very powerful in terms of, especially for the mRNA vaccines. you know it just showcases how we can respond to emergent threats from different viruses in a rapid way. Beyond prevention, of course, they can play different types of roles, especially in reducing viral transmission evolution. So they weaken the virus, and it's a way of preventing an infection, and of course, it leaves the immune system always alerted against different viruses in case of infection or another infection in the future. As you can see here, we have different types of vaccines that are produced, that are made. DNA vaccines, we said mRNA vaccines, nanoparticle vaccines. There are vaccines that are based on the vectors, the vector vaccine, other vaccines also, subunit vaccine as well. They can be based on whatever, unless they do the job and they, get to the right target. We're gonna talk now about the personalized medicine. Personalized medicine is just, it's the future. It's just, it's something that researchers are working on, and it's the future of antiviral therapy, because we don't know the organism. We don't know, how every organism works, how our immune system or the immune system of each person works. So we need to have a map of the, for example, the microbiome of the person. We have to know, the diet. We have to know the immune system and how it works. We need to map the organism, as a whole, in terms of either molecular, at all aspects, genetic, etc. So we can actually develop a, you know, a therapy that is specific, and just specific just for that person. So the future of antiviral therapy lies in personalization by integrating genomic and immunological data. So yeah, we have to know all the informations that we can gather on the person to be able to treat him. Treatments can be tailored to individual patients, improving efficacy and reducing adverse effects. You know, it's just going to be very specific. It's going to be, predictive, preventive, and personalized at the same time. So we can, as you can see here, we can prevent, different types of complications, and we can predict also different, the risk assessments. We can predict patient stratifications, the prognosis, diagnosis, etc. We can also here personalize. We say personalization, dependent on the profile of the person, and also targeting individual molecular pathways of the person. So it depends, and it helps also. It's very costly. It's very, very costly. It is applicable, but it's very, very costly. And as I said, it is something that's personal. It's something that works just for one patient, and on one patient only, as we call it, the personalized therapy or medicine, right? So we have looked upon different types of therapies, regarding the antiviral drug resistance, and why these viruses, why they resist, and what we can do, using different therapies and technologies to unravel these challenges. So we're left with the challenges now and the future outlook of these methods and these approaches. So, despite all the advances that we have seen in research to combat viruses and to combat the antiviral drug resistance, challenges still remain. You know, as every scientific question, as every scientific approach, we still have limitations. So these include high costs, of course, as I was speaking, throughout every therapy approach, we're talking about high costs, limited accessibility. Of course, not everybody has access to these types of therapies, even if you have the money. Sometimes, it's limited. Ethical consideration, absolutely, particularly with technologies such as gene editing. It's still something that's very niche, that's in the works. We can't just like apply it to a person. Even if we have the information, we have everything. It still has to undergo a research and confirmation and validation, of course, of these approaches. Addressing these issues will be essential for translating innovation into real world impact, as we said, and to be translated into clinical practice. As future outlook, so looking ahead, the future of antiviral medicine will rely on integration. We absolutely have to integrate every approach we can have by combining, for example, AI. We talked about genomics, the immunology, immunotherapies, nanotechnology. So, with all of this, we can move ahead. We can move ahead towards predictive and preventive strategies that stay ahead of viral evolution. So, research remains, of course, it remains a key aspect to unravel these challenges and these future outlooks regarding this antiviral therapy. But combining all of this, as we have already said, the combination therapy is key. As we develop a very high specific working sensitive therapy that can be robust at the same time and have a rapid effect on these viruses, and to maybe in the future outmaneuver them, even. So I will conclude, I think, today's presentation. Yeah. So to conclude this presentation, it's a very interesting subject. So drug resistance is an inevitable aspect of viral biology. It's a very concurrent challenge. As with also the antibiotics and antibacterials. The bacteria that is resistant. These microorganisms are always, they're very powerful, and they're always predisposed genetically. They have these systems. It's just the selectivity that makes the difference, and also the control. So just looking to control these infections. However, through innovation and interdisciplinary approaches, so we have to make call, to all these disciplines, all these fields to come together, develop strategies that allow us to anticipate, manage, and potentially overcome these challenges someday. So the future of antiviral medicine lies not only in new drugs, antiviral drugs, but in smarter and more adaptive systems. So we need something that can go for a long time. We need something that's specific, that is kind of smart, because as we know, viruses are very smart. They're the smartest microorganisms. So you have to understand how they manipulate, how they move, in order to outsmart them, and create a system that can go for a long time, can go for long, and be adapted to different cases and different viruses and different systems as well. uh, you know so I think that's going to be it for today, for the presentation. Thank you very much for your attention, and I would be happy to take any questions. So yeah, thank you.
Thank you so much, Dr. Mona, for such an insightful presentation. Please let me confirm, is I audible to you?
Yes, yes, I can hear you. Thank you.
Okay, okay. Thank you, thank you. It was truly enlightening and gave us a deeper understanding of the future directions in antiviral medicine. Now we would like to open the floor for the question and answer session. I encourage all
Of course.
participants to actively engage and take this opportunity to ask your questions.
Yes.
You can type your questions in the chat box. Kindly keep your questions brief and relevant so that we can accommodate as many as possible. Uh, and the floor is open for questions. Participants, please go ahead.
Okay, thank you.
Please type your questions as soon as possible. Uh, there is a question in this chat. So, Miss Mona, should I read for you or you can read?
Yes, please. If anyone has
Okay.
any question or any suggestion, please can leave it in the chat, or you can activate your mic and just ask me.
Actually, participants can type in the chat and the question is highlighted if you can read. Otherwise, I can read for you.
Okay. I think I see a question. So, Mr. I think Mohsin said, what's the most promising strategy right now to actually prevent resistance from developing in the first place? Great question, actually. And and it's very insightful that you have asked this question, as I have shown. I think there were five strategies, I think, that are that were all very strong candidates to be promising for the prevention of resistance. So, as for my personal overview on this, I think, uh, as you've said, to prevent resistance from developing it from the first place. So we have seen the monoclonal antibodies, as part of the immunotherapy. I think they are strong candidates, of course. to prevent the resistance, as they act upon the receptors of the host cell, and they prevent entry to the host cell. So we won't have a replication, and therefore there's no protein assessment from the virus. So I think it's immunotherapy in general, and we're talking specific. We have the monoclonal antibodies, as a very, very promising strategy nowadays. Yeah.
Thank you so much, Doctor, for the answering. Here is the next question you can see in the screen.
Yeah. So, yes, another question. How we use antiviral medicine in a simple and modern technology era. In a simple and modern technology era. So, I think the antiviral medicine, it is used, you know, it is used today, in modern times. And, when I think you're bringing up the technology part, I think it's just, a matter of choice. So, as I said, the technologies that I just brought up, they are in the works. Some of them are applicable today. But in terms of use, depends on the person, depends on the case of infection, as well as the cost of this therapy. So, for example, we have the CAR T cell therapy, which is an immunotherapy it is applicable clinically nowadays. It's very costly. It is, advanced in terms of technology, but it is applicable, and you can use it freely, of course, if you have the cost. And of course, in leading establishments, healthcare establishments worldwide. Yeah. Do you combine therapy is equally effective for all? uh Effectiveness, it's very, very great question also. So when we talk effectiveness, we talk about your immune system. So we have different built and different immunological systems. So when we're talking about effectiveness here, it depends and it differs from one person to another. And also it depends on the infection. Like, are you infected with just one virus? Or, for example, do you just have, the Zika virus? Or do you have, for example, a double infection or a triple infection? So it absolutely depends on the case. It is very effective, of course. You're asking about effectiveness, but it's not granted, dependent and on each person and their immune system, and how do you react to the combination therapy. Okay, we have another question. What are the most promising strategies to overcome antiviral drug resistance in the next decade? A great question. So I've talked about in the next decade, the future. We have AI. We have artificial intelligence, in the service of antiviral drug resistance. So absolutely, AI is very uh, a robust tool, and it's revolutionizing already the viral drug discovery. It is very, it's helping. The development and the research of different antiviral drugs. So I'm thinking, in my opinion, we can combine the AI therapy or AI artificial intelligence, discovery, antiviral drug discovery with another type of therapy. Can be a very, for example, CRISPR. The CRISPR gene editing can be very powerful with AI. Yeah. How these nano carriers can work, how we can use nanotechnology, how we can take these nano carriers, and what is their method of using. So these carriers, they are, it's a type, it's an engineering, you know, it's a buildup. It's something that is very, it's a wet lab work, in and we use also the informatics part, of course. So they are made of, depends if you want, the target, you want DNA or you want RNA. So you just build an environment that can, of course, transport, for example, a drug that is DNA based. So you have to deliver DNA to the target. And, it's a different types of, for example, we have proteins, we have enzymes, we have different types of carriers. We have even DNA and RNA can be carriers. We have lipids. We have, it's dependent on what you want. And of course, the carrying is done by the substance that you have made. From the very start, it's built up nanotechnologically. With microscopes that are very, very, high end. And the buildup is very the building up, sorry, is very specific. It's sensitive. And it's an engineering. So it's not very it's not easy. Okay, how can developing countries contribute to antiviral drug research? Developing countries, well, of course, it's critical in terms of course, the lab equipment, the financial, contribution as well. It's very demanding. I'm not gonna lie, it's very demanding to do research on the antiviral drug resistance. We need, very equipped labs, and labs that are following the ethical rules, and that are very equipped, and are also because we are handling viruses, so it needs to be very, very secured as well. So yeah, it's a bit of struggle, but it can happen, of course. Can AI predict the pattern of resistance of any virus in response to any specific kind of drug? So we're talking about patterns of resistance. This is the key point, actually, of AI, as we've talked about the targeting. So this is the right use of AI in this the response, to any specific kind of drug. It's what we aim to do. It's what we aim to discover using AI. It's just like bioinformatics. When you find the target, for example, between the proteins, and their attachment, look for the substrate, and it's the same thing, but it's a little bit more specific, and it's in the works. Different labs around the world, they are working on stuff like this. And of course, if it's done the right way, and if it's developed the right way, we can actually predict these patterns, and in any antiviral drug, of course. Can we use nanoparticles as antiviral medicine? Aren't they lethal for humans or animals? We talked about the toxicity effect, of course, of these nanoparticles, but on on the fact that these nanoparticles and this nanotechnology, they can protect actually the fragile molecules. In case of RNA, they improve, the targeting to specific tissues, and they reduce systemic toxicity. So we're not talking about the nanoparticles that actually damage or harm the humans or the animals. Okay, so the nanotechnology that enhances antiviral therapy is primarily done through improved drug delivery. So we use substances that are not harmful to the body, and actually we build and we engineer delivery systems that are from our body, so we can find protein, lipid, DNA, RNA, microRNA, RNA messenger, etc. And we build the nanoparticles ourselves, and the delivery system is primarily it's not harmful at all. What skills and research areas should biotechnology students focus on to build a career in antiviral drug development? Great question. Amazing question, actually. As I've already brought up, the multidisciplinary side of this area of research. So I think, in my opinion, molecular biology, of course, is a must. Of course, also genetics. Genetics also is very critical. The understanding of mutations and all of that. Also, immunotherapy, immunology in general, is also a very indulging discipline. These three are the top three disciplines that should be uh, up there, for the learning and the career, of course, for antiviral drug discovery and resistance. Of course, the virology is a no-brainer for this kind of this kind of career. Virologists at first, and then of course you branch out to different disciplines, or you can of course study them at the same time. In terms of, for example, the master's degrees, they have these disciplines, of course, aligned with the programs. Yes. Any other question?
Thank you so much, Dr. Mona, for addressing each question very clearly. I think participants, had you any other question, you can type, because further I did not see any question in the chat. If you have still any question, we are here waiting for 1 or 2 minutes. You can type your question in the chat.
Yes, of course.
I think all questions and queries are addressed, because Dr. Mona addressed it very clearly. And so, should we end now?
Yeah.
Okay. Dr. Mona, thank you so much for being here and giving such an insightful presentation and addressing each question. And I am also thankful to the organization Vitanova International Alliance for Sciences for organizing such sessions, and also thankful to our host, Dr. Shahzad Zafar, Senior Vice President, Vitanova International Alliance for Sciences, for being here.
Of course. Thank you so much for today. It was a very great learning experience. It was a great webinar. Thank you all, for the Deputy Director, of course, and the Vice President of Vitanova International Alliance for Sciences. Thank you so much for this opportunity. Thank you for the questions. And yeah, thank you so much.
Yeah, thank you so much, Doctor. And I am also thankful to the participants who stayed in the session till the end and managed their time. And I am also thankful to the whole team behind this session to organize and putting their efforts for the students and for their well-being and for their future. And thank you so much, everyone. So now we are going to end. If you have still any queries, you can send me in the WhatsApp, so I can discuss with doctor and give you the answers. Okay, thank you so much.
Yeah.
Mr. Mansoor, you can end the session now. Thank you.